Hey everyone, my name is Riley Jaekel, and my presentation today is going to focus on cocaine and its effect on the neurotransmitter dopamine. So let's do a little refresher on dopamine. Dopamine is involved in how we feel pleasure. Dopamine's functions include executive function, motor control, motivation, arousal, reinforcement, and reward. Dopaminergic neurons are cells that produce dopamine within the brain. These cells are located in the substantia nigra in the ventral tegmental area in the midbrain, and within the arcuate nucleus in the hypothalamus. After the production of dopamine, it is packaged into a synaptic vesicle and stored until action potentials induce the release of dopamine into the synaptic cleft and cause binding to dopamine receptors on the postsynaptic neuron. The binding of dopamine to the receptor initiates a cascade of signaling responsible for activating functions in specific areas of the brain. Once produced, dopamine can travel through different pathways in the brain. A major pathway, the mesolimbic pathway, goes from the ventral tegmental area to a region called the nucleus accumbens. This pathway connects the dopamine-producing area with a major reward center. As dopamine is produced in the ventral tegmental area, it is then released in the nucleus accumbens, where dopamine levels regulate the motivation and desire for rewards. When dopamine levels are high in the nucleus accumbens, people feel satisfied, fulfilled, and motivated. For example, when someone receives a reward for something they've done, dopamine levels continue to be high, and the feeling of satisfaction and fulfillment is extended. All feelings are in some way connected with the feelings of reward and pleasure. We all get accustomed to a certain level of dopamine in this part of the brain during our lifetime. Because of that, the dysfunctionality of the mesolimbic pathway and the subsequent decrease in the dopamine levels can force us to seek the feeling of reward from other sources. Simply put, when there's not enough dopamine in the mesolimbic pathway, the brain tries to make up for the lack of this neurotransmitter and to stimulate the neurons responsible for pleasure and rewards. When the production of dopamine and the functionality of the mesolimbic pathway are deregulated, people can develop addictions to things such as cocaine or other disorders. This is why people become addicted to gambling, drugs, sex, or any other activity that generates high dopamine levels and that intensive feeling of satisfaction and pleasure. So, cocaine is a commonly abused drug that also goes by the name of coke. Derived from the coca plant, cocaine is characterized as a stimulant drug. Stimulants activate the central nervous system to produce arousal, increased alertness, relieve fatigue, decrease appetite, and elevate mood. Cocaine most commonly comes in the form of a fine white powder, but its cracked counterpart comes in crystallized rocks. Cocaine can be used a multitude of ways, but it's most commonly snorted or smoked. In a survey conducted in 2016 by the National Institute of Drug Abuse, 1.4% of 8th graders surveyed had tried cocaine in their lifetime, while 0.8% had used the drug within the past year and 0.3% in the last month. In the graph to the right, you can see that cocaine use increases with its peak of users 18 to 20 years old, and then it decreases into the adult years. Under normal conditions, dopaminergic neurons regulate the amount of dopamine and produce it at appropriate intensities for certain functions. However, with cocaine present, it interferes with this latter control mechanism. Cocaine ties up the dopamine transporter, a protein that the dopaminergic cells use to retrieve dopamine from its surroundings. As a result, dopamine molecules that would otherwise be picked up remain in action. By blocking the reuptake, dopamine molecules begin to build up and overactivate the receiving cells. Cocaine can also inhibit the transporters of other neurotransmitter chemicals, like serotonin and norepinephrine, but its actions on the dopamine system are generally thought to be most important. Cocaine blocks the reuptake of dopamine anywhere in the brain that has dopamine transporters. Cocaine's ability to produce euphoria is due to its effect on the limbic system. This system is highly concentrated with dopamine-responsive cells. When affecting the limbic system, the nucleus accumbens seems to be the most significant site. The natural function of increased dopamine within the nucleus accumbens is to keep us focused on activities that encourage the basic biological goals of survival and reproduction. When cocaine causes a buildup of dopamine in the nucleus accumbens, it creates an extreme feeling of pleasure. After a single dose of cocaine, the amount of dopamine connecting to receptors in the nucleus accumbens can exceed past amounts produced during natural activities. With this being said, cocaine can produce feelings of pleasure greater than pleasure felt after thirst quenching or even sex. Cocaine also has an effect on the brain's memory centers located in the amygdala and hippocampus. These memory centers help us remember activities that we did that led to pleasure associated with an increase in dopamine within the nucleus accumbens. During a cocaine high, a person's memory center imprints memories of the intense pleasure. This memory also includes the people, places, and objects associated with the drug. Due to this, going back to a place where cocaine was used or just seeing cocaine-related media can trigger memories and the desire to repeat the experience. Delta-phos-B is a pace-setting chemical that occurs naturally in small quantities in the nucleus accumbens. It is characterized as a transcription factor because delta-phos-B remains in the cell that produces it and stimulates certain genes. Researchers believe that the effects of cocaine on delta-phos-B may constitute the transition from drug abuse to addiction. Due to delta-phos-B's extreme stability, a single molecule lasts for 6 to 8 weeks before breaking down. Each new episode of cocaine abuse heightens the buildup of the molecules that has accumulated from previous episodes that occurred within the last 2 months. In a daily user's case, delta-phos-B will remain extremely elevated all the time. In studies with transgenic mice, those with elevated levels of delta-phos-B exhibited behaviors that correspond to human addictive behaviors, while the control mice did not. Delta-phos-B helps to determine the basic properties of cells, including its structure. While cocaine affects several transcription factors, its effect on delta-phos-B appears to be the most long-lasting. Like dopamine, chronic exposure to cocaine causes an accumulation of delta-phos-B in the nucleus accumbens. In the first graph, you can see that delta-phos-B remains stable for several hours compared to other proteins in the phos family. When you look at the second graph, this shows how cocaine slowly builds up the amount of delta-phos-B in the nucleus accumbens, and levels remain high for quite a period of time. It is presumed by scientists that delta-phos-B is responsible for more than 25% of all chronic cocaine-induced changes in gene expression within the nucleus accumbens. Cocaine abuse and addiction does have some clinical implications. While delta-phos-B is a highly stable molecule, it does not explain why former cocaine abusers often relapse after months and even years of abstinence. Due to this, cocaine must have long-lasting neurobiological effects. Scientists have discovered alterations in the nerve cells located within the nucleus accumbens. Chronic exposure to cocaine is causing cells to extend and produce new offshoots on their dendrites. Dendrites are branch-like fibers that extend from the cell body of a neuron and receive incoming signals from other nerve cells. With more dendrite branches in the nucleus accumbens, a greater volume of nerve signals from other regions, such as the hippocampus, amygdala, and prefrontal cortex, will be collected. This would give the other regions an increased influence over the nucleus accumbens. In turn, this may cause the long-term behavioral changes associated with cocaine addiction. It is unknown how cocaine causes the offshoots in dendrites, but it is assumed that delta-phos-B is involved. CDK5 is a gene whose function is to regulate nerve cell growth, and it is stimulated by delta phos-B. From what scientists know about delta phos-B in conjunction with cocaine abuse, they predict that the increase of delta phos-B activates the overstimulation of CDK5, which causes the dendritic sprouting. The increased offshoots on a person's dendrites plays a role in the behavioral changes we most commonly associate with addiction. Due to the increased signaling towards the nucleus accumbens from areas such as the amygdala, hippocampus, and prefrontal cortex, an influx of dopamine has been observed. This may be a problem, as we know increased dopamine in the nucleus accumbens gives cocaine that euphoric feeling that can become addicting. Memories associated with an increase in dopamine in the memory centers will have a larger influence than they did before due to the dendritic sprouting. Lastly, increased signaling from the prefrontal cortex will cause more poor decision-making and erratic behavior from the cocaine abuser. A study conducted in 2012 analyzed the effects of acupuncture on stress-induced relapse to cocaine-seeking rats. The rationale for this experiment was that exposure to a stressful situation can lead a former cocaine abuser right back into addiction, and it is considered a major risk factor for relapse. Neural substrates associated with stress have been seen to have a great impact on the mesolimbic pathway of dopamine. Stress increases dopamine release in the nucleus accumbens, which then reinstates drug-seeking behavior. This suggests that stress affecting the release of dopamine in the nucleus accumbens leads to relapse. Male Sprague Dawley rats were trained to self-administer 1 milligram per kilogram of cocaine with a lever for 14 days, and then they were subjected to 15 minutes of intermittent exposure to foot shock. Foot shock is performed by the rats standing on an electrified grid that can produce shocks of different intensities. The rats then received 1 minute of acupuncture to their Shinmen acupoint on the inside of the wrist directly after the foot shock was terminated. The other test groups consisted of rats that received foot shock and then given acupuncture to the Yanghai acupoint as a control. In the results, the rats that only received foot shock reinstated their cocaine-seeking behavior. In the rats with acupuncture applied to the Shenmen acupoint, a decrease in cocaine-seeking behavior was observed. Cocaine-seeking behavior within the rats was reduced by 86% with acupuncture performed directly after the foot shock induced stress. A significant reduction in responses on the lever was also observed when acupuncture was applied. Cocaine-seeking behavior remained the same with acupuncture applied to the Yanghai acupoint. Due to these findings, the hypothesis that acupuncture can prevent stress-induced cocaine relapse was proven correct. However, this decrease was only seen when acupuncture was applied to the Shenmen acupoint, suggesting that acupuncture's effect on cocaine relapse is site-specific. The next step researchers plan to take is to look at acupuncture and how it may affect dopaminergic receptors in the brain of those with cocaine addiction. In conclusion, dopamine is a significant neurotransmitter that is produced in several regions of the brain and is linked to how we feel pleasure and reward. Cocaine is a stimulant drug that acts on the human brain by blocking the reuptake of dopamine, mainly within the nucleus accumbens and the major memory centers, the amygdala and the hippocampus. Cocaine's effect on dopamine makes it a highly addictive drug, with former cocaine users going on years of abstinence still relapsing. Delta phos-B is a transcription factor that is highly stable and may hold the switch from cocaine abuse to addiction. Cocaine causes dendritic sprouting that may be the cause for long-lived behavior changes associated with addiction. Lastly, due to scientists' findings, acupuncture may be an effective treatment in decreasing the dopamine built up from stressful situations, thus decreasing stress-induced relapse.